So we've finally gone through the first step of the central dogma, transcription, and we have our mRNA. So the second step of the central dogma is to turn that mRNA into protein using a process called translation. Translation relies heavily on the molecular machinery of a component known as a ribosome. Ribosomes are the main driver of translation. Ribosomes are actually able to read an mRNA sequence by taking it in at one end and reading the mRNA sequence all the way through to the other end. And as it does this, it takes those 20 amino acids and reads the code determining which amino acid belongs in a particular position. Now, one of the most exciting puzzles of the 20th century was to figure out how this code worked. I mean, how is it that this genetic code, that these 4 letters, and in what combination is it that the ribosome can read this and add the right amino acid? Was it one base, one letter per amino acid? If true, that would only give us 4 possible combinations, an A, a G, a C, or a U, and we know that we have at least 20 amino acids to get to. If it's 2 bases per amino acid, then that would only be 16 possible combinations. Things like AG, AC, AU, GC, GA, GU, and so forth and so on. Only 16 combinations, but still 20 amino acids. So just extending that logic a little bit further, what's the minimum number of bases needed to have enough combinations to code for at least 20 amino acids? Go ahead and put the number that you think is best in this text box here.